So today we will talk about the autosomes. Autosomes and sex chromosomes. Autosomes and sex chromosomes. Basically, autosomes are the chromosomes which do not determine the sex of an individual. Basically, chromosomes that do not determine the sex of an individual are known as autosomes, right? In an human body, like what are then sex chromosomes? Sex chromosomes are the chromosomes which determine the sex. They determine the sex, and autosomes are the chromosomes which do not determine the sex of an individual. So in human, like if we talk about, we have total 46 chromosomes. So out of them, 44 are the autosomes, right? Or you can say 22 pairs are the autosomes. These autosomes basically determine the somatic characteristics, right? They are responsible for somatic characters, right? If we talk about a female, so it will be having 2n plus is equal to, obviously it will be diploid, then it will be 44 plus XX chromosomes. Or if we talk about the haploid condition, then 22 plus X, 22 plus X. If we talk about male and it is in diploid condition, then it will be 44 plus XY, right? XY chromosomes. And if we talk about the haploid condition, then it will be having 22 plus X and here 22 plus Y, right? But in some organisms, basically the position of the position is reversed, right? For example, in fishes, in moth, basically in birds, the position of XY chromosome is basically replaced, right? Now what happens if it is a female? It will be having ZW, right? If it is a male, then it will be having ZZ, like this. So like this, the position is basically replaced in some organisms, right? So now let's talk about the karyotype. Basically, karyotype is the picture of the chromosome, right? It is a picture of It is a picture of chromosome of an individual species. Of individual species, right? And it is basically used to determine the gender. Or basically to determine the abnormalities. Right? For example, let's take a blood sample. And this sample will be going through the process of centrifuge, right? And then we will be treating it with a chemical known as colchicine, right? Colchicine is basically a chemical which basically prevents the formation of spinal fibers. Right? Now when the slide is prepared, now basically what will happen, as we said, that after treating these blood sample with this colchicine, consider that here we have this karyotype and here we have chromosomes, right? So during the metaphase, we know that the chromosomes come in the center, but when they'll line up in the center, but there won't be the formation of spinal fiber, so how they will be stretched? So at that time the picture will be taken. So that picture is this one, and this picture is known as ideogram. Right? Basically, we have two types of, like, if we see, if by looking into this ideogram, if we see if there is 3 chromosomes on 21th pair, so if it is 22n plus 1 to 1, so it will be equal to 47. So this type of disorder is known as trisomy. Right? If on the 22nd position, if there is 1, like if it loses 1, then if it is 2n minus 1, then it will be 45. So it is known as monosomy. So basically these are the chromosomal aberrations. It means the change in the number of chromosomes. Right? But there here can be structural aberrations as well. Means the change in the structure of the chromosome is known as structural aberrations. Right? If we talk about the karyotype, there are basically two types of karyotype. Right? Symmetrical. Other is basically asymmetrical. Okay, symmetrical is the one in which the ideogram basically metacentric and submetacentric chromosomes are determined. Right? In which metacentric and submetacentric chromosomes are determined. Right? But in asymmetrical one, there will be the different chromosomes. There will be different chromosomes. Right? So I hope this is clear to you. If you have any kind of queries in the autosomes and the karyotype, you can ask me in the comment section. I'll be really glad to answer you guys.